Good morning, everyone, to the next day of the Advanced in Silico Drug Design workshop and challenge. I will have the opening session, and it's about very basic but very important topic, and this is molecular similarity as well as its usage in optimization. By way of similarity, it's a good way to introduce a few, I would say, fundamental concepts of molecular representations, the molecular descriptors, ways in which we can describe our molecules. So I will start with similarity. That's the first part. The second part will be apply part, or let's say a bit more general part, and this is about optimization. So first, well, it actually is similarity. It's actually very vague concept. As it's remarked here in a book from the 1970s, it's not about chemistry. It's about, I think, literature analysis. This author says everything is like everything in endless ways. So he remarks, okay, my pony, it's like a horse, but a little bit smaller. The floor is like the earth, but a bit more local, and so on. So if you can find between two things, if they are different, you can find similarities between them. So the question is how to find meaningful similarity. Okay, here are a few more definitions. What's similarity? It's a degree of sameness for different things. So let's say shared features, shared properties of different things, or non-identical things, we can call them as well. So first something to think about. So which of these three molecules are the most similar to each other? I'll do the thought process out loud. So let's say the most straightforward thing that we could say is, well, we have this tropane skeleton in the left one, and we have this tropane skeleton in the middle one, and we don't have it here. On the other hand, if we imagine the molecules in 3D, we could say, okay, this molecule here, it has an ester in there, methyl ester in there. This molecule here has a methyl ester in there. And maybe if we have good spatial insight, even, okay, we can imagine in 3D how well this will overlap. So, okay, now I will reveal what these molecules are. You can see the first one, that's atropine, that's anticholinergic. It's a typical ingredient or plant component that you can find in nightshade plants. It's known to be very toxic. Sometimes young children eat the small berries and they get poisoned from it. On the other hand, these two that don't instantly look that similar, actually they work in the same way. One is cocaine, recreational drug that's stimulating. The other one is methylphenidate, also known as Ritalin, and they both work via dopamine reuptake inhibition. So what's the reason why? How come the structural similarity that we see here in a superficial way, or let's say in an instant visual way, how come it doesn't lead to the pharmacological similarity? This is because of something like a pharmacophore concept. So the features that are important for binding, aromatic ring, basic nitrogen, and an ester in the right place, apparently they overlap in a spatial way better in these last two examples. So just as a thinking experiment, we can conceive of similarity in more than one way. And depending on what's the outcome that we want, actually these ways can be really different or even opposed. Now, why do we care about similarity? The main reason why we care about similarity in chemistry or medicinal chemistry context is because of what is called the similarity principle, or outside of chemistry, this is also called neighborhood behavior. And this simply means when you have a similar structure, you expect similar properties, including biological properties. For example, here there is a very often quoted number. It's a little bit bad rule of thumb, but TS, it stands for Tanimoto similarity. I will explain later on what this means. In this specific case, it's of a daylight fingerprint. It's not so important which one it is, but basically it says once you are over a certain amount of like structural chemical similarity, then you may expect the same biological activity. Of course, in practice, it's not always true, but as a rule of thumb, nonetheless, it's often remarked. Another reason why we care about similarity is because of the applicability domain problem. So if we are building models, if we are making predictions in whatever ways, we have more confidence in our predictions when we have more similar data in the input set, in the test set that we are trying to predict. And this means we can use similarity to judge how confident can we be in what our models say. And then finally, in general, the whole endeavor of ligand-based drug design, so drug design that begins with the ligand, this is based on finding common features, and this means this is based on similarity. So if we want to do a ranking of molecules, we can do this similarity-based. If we want to do clustering of molecules and so on, we must use some kind of smart similarity metrics to do that. Yeah, and just to remark, if I think probably yesterday it was made clear, but just to repeat it, basically in drug design you have two approaches. The first one is the ligand-based approach, which is the one we are rather talking about today. And there we use only information about the ligands itself, the information contained in the ligands. We don't use explicit structural information like some PDB X-ray structure or something like this. In SBDD, structure-based drug design, we will rather use this. Or, for example, we will model these structures by homology modeling or AlphaFold or whatever. Okay, yeah, as we saw in our example, molecules, they can be similar in more than one way, and choosing meaningful features, this is really very, very crucial to make a similarity scoring endeavor fruitful. So in what ways can molecules be similar? Well, there are several ways. I list here a few basic ways. So the first way to consider it is topological. It's based on atom connectivity, is based on considering the molecule as a graph. So let's say a graph where the nodes are the atoms and the edges between the nodes, they are the bonds. We can consider this in two ways. First of all, local. So this is the presence or non-presence of substructures, functional groups, atoms, and so on. Or globally, where also we care about the topological distance of substructures, atoms, and so on. So topological distance, in this case, it simply means the distance expressed in bonds, not geometrically. Then the next type of similarity we can talk about, that's geometrical similarity, and this is based on molecule geometry, and this means that we need coordinates. Usually 3D coordinates, in principle, it can be 2D coordinates as well, but it tends to be more meaningful and more corresponding to reality to use 3D coordinates. And in that case, for example, we can calculate the Euclidean distance between, again, substructures, functional groups, atoms, and so on. We can calculate various indices to see the shape similarity between two molecules. We can have a look at the electrostatic surface. We can have a look at the partial charges and so on. Or we can go even a little bit more abstract, and we can see if we have pharmacophore matches. So this just means if it matches to a certain 3D distribution of features. Then another way in which we can compare molecules is physicochemically. So this means that we look at physical chemical properties. It can be things like polarity, even molecular weight, such things. Importantly, this is actually something which can be measured. Let's say something like a partition coefficient, we can measure this. Then another way in which molecules can be similar is biologically. This is something that we can predict. We can predict some activity using QSAR. This will be talked about later today, or pharmacophores, or whatever model we try to build. It can be measured as well, of course. But on the other hand, often when we are doing similarity-based experiments, this is the endpoint. This is the thing that we want to predict. And let's say we want to, using similarity, this is rather what we want to have as our endpoint, not as our input that we use to score our similarity. Now, a bit less abstract, or a bit more concrete. Let's talk about how exactly we describe these features. And the way we do that is through descriptors. And here, actually, there is quite a nice conception of what is a descriptor. You can compare a descriptor as something like a keyboard. which you would use in searching library of books or some database more generally. And that's actually one of the main uses of descriptors and fingerprints outside of biological activity prediction, searching chemical databases efficiently. So what type of descriptors exist? Actually, there is a huge amount of descriptors, any kind of, let's say, meaningful feature that you can calculate, predict, or measure for your molecule. You can count for, you can count as a descriptor in the widest sense. So some very typical ones are the presence or non-presence of substructures, or one substructure out of a pool of substructures, the polarity or the partition coefficient, maybe predicted toxicity, for example, predicted with a simple QSAR model, certain graph invariance, for example, if you calculate the eigenvalues of your adjacency matrix, those you can use as descriptors. You can put measurements in there as well. For example, high throughput screening measurements. If you measure it for your whole set of interest, you can use this as input data to do more advanced modeling. You can have various 3D features. Again, you can use pharmacophore matches for this. Then you can also use substituent contributions, so typical like Hammett constant type factors. Now, descriptors can be used to construct fingerprints, or rather, a fingerprint. It's an efficient and standardized representation of chemical features, and each of its elements we can consider to be a descriptor. Of course, if we have descriptors, we don't need them to construct a fingerprint. And very often they will be bespoke descriptors that we will need for whatever purpose. But typically, if we are building models, if we are scoring similarity, we make use of fingerprints specifically because they have this nice standardized representation. So, for example, very typical form, that's the binary vector that has a fixed length. So let's say, for example, a vector of, I don't know, 1024 bits, something like this. And because of the standardized formats, it means that we can use quantitative similarity metrics. So some typical examples of this are the extended connectivity fingerprints. I think this is one of the most used ones for simple, straightforward QSAR models. I will explain in detail actually how this one is constructed. Another one, that's the structural keys. I will have short illustration as well about these. And then there are various more types. There are atom pair fingerprints that encode shortest paths between atoms or substructures, depending on how you set it up. There are various types of pharmacophore fingerprints. There are really many types, and usually you choose them according to the task which you want to do. As I said, the reason why we are interested in it is because of building models, estimating similarity. Another reason why they are useful is for efficient searching. And for example, specifically this max structural key. The reason why it was made is actually not for building models, not for even estimating similarity, but it's rather for making the searching of chemical databases like in like SciFinder or Raxis to make this a bit more efficient. You can see here on the left an example of a max key, or rather a part of a max key. Normally its length is 166 bits, as compared to on the right an ECFP, an extended connectivity fingerprint. So, so what's encoded in there? In the max case, this is something which we call a structural key. And this means that every bit in our bit vector, it corresponds to the presence or non-presence of one discrete substructure type. So for example, in this case, it would be this bit, it stands for benzene ring with one substituent. This bit stands for an amine, and this bit stands for carboxylate. Actually, some of the bits, they are a little bit more abstractly defined. So there will be things like a nitrogen with a 2-atom linker to an oxygen, those type of things. But still, in the end, the important thing there is one bit has one meaning. The ECFP, it's a little bit more involved, but this is actually what makes it more powerful. And here, what you will have is rather in a generic way, you calculate identifiers for substructures at a given radius, and then you fold those into a fingerprint. And this means that one bit, actually, it can correspond to a whole wide array of fragments. So let's look at this in a little bit more detail. Let's look at the construction of the ECFP, and I have here this example molecule, O-methyl proline. So first of all, let's say explicitly, what am I talking about when I mean radius, substructure, environment, neighborhood, and so on. So when we construct these substructures, we center them on an atom. And we do this on every single atom in the molecule, so 0 to 8, all 9 of them. At radius 0, our fragment, that's simply the atom. So carbon, oxygen, carbon, oxygen, carbon, nitrogen, and so on. When we extend the radius, this means that we include all direct neighbors of the radius 0 fragments. So, for example, atom 0 has as its neighbor atom 1. Atom 1 has as its neighbor atom 0 and 2. Atom 2 has as its neighbor 1, 3, and 4, and so on. And in this way, we construct our radius 1 environments. Then for radius 2, actually, we do exactly the same thing. So we consider the fragments we constructed in the previous step, and we include all neighbors, which just means increase the size, the topological distance with 1, and then we have these fragments. So now the question is, how do we get this folded into a nice fingerprint of given length? Well, for this, we have hashing and the Morgan algorithm. So what do we hash? How do we assign a number to each atom? Basically, first we make a list or an array or a tuple or whatever with the atom properties that we care about in them. So in this example shown, what we care about is the atomic number, 6 for carbon, the degree, this means 1 outgoing bond, and the amount of hydrogens. It's methyl, so this means three hydrogens on there. Okay, I assign this for all of the atoms. For example, this one is oxygen, 2 outgoing bonds, 0 hydrogens, and then we apply our hash function. And actually, this hash function, it can be whichever you want it to be. Typically, there is one chosen that gives a 32-bit integer as its output. It should be broadcast ideally, totally randomly and evenly over the 32-bit space. And what this now means is that each of these atoms, they have a number associated with them. Yeah, so I put them here above. And now the question is, how do we assign these numbers to our environments, to our substructures as well? So what we do is we make again, we make a list or array or tuple with the neighboring atoms identifier in there. So we put this number in there, we hash it, and then we get an identifier that corresponds to a given fragment. And this means that actually beforehand, we don't have to assume anything. We don't need to know about any functional groups. We don't need to know about what is common in our training set or anything like this. It's very generically defined. But there is one more step, and this is we need to fit it in a fingerprint of a given length. So here it's shown as a fingerprint of length 64. And the way we go from this bag of numbers corresponding to a bag of fragments to a fingerprint is we apply a modulo operation. So we do modulo 64. So we get a number between 0 and 63. And this is simply the index in the, so we just turn on a bit. If that number is in there, let's say 9 is in there, for example, then we turn on bit number 9. If, okay, we fold this number, we get 19. This means we turn on bit 19. And with this in hand, we have our ECFP, we have our extended connectivity fingerprint ready for doing our modeling. Now, with this in hand, now we can start to estimate similarity really quantitatively. So not just, okay, how we feel about it, what looks similar to our eyes, what is nice, but let's say assign some numbers to it so that we can talk about it a little bit more objectively. And by far the most common way to do this is by Tanimoto similarity. And a nice way to think about this is features in common divided by the total features, or as it's shown here, on this Venn diagram display, it's as simple as you get the union of features. Let's say A is the bag of features in molecule A, B is the bag of features in molecule B. So you get the amount of common features on top, and you divide it by the total amount of features. So quite straightforward. But now imagine that as our descriptors, we chose something continuous, then we can still calculate the Tanimoto similarity, but it's a little bit more difficult to imagine. What's a bit easier to imagine is to use typical distance metrics. The most common one would be just the Euclidean distance. So we can, let's say, imagine them as points in Euclidean space of dimension, whatever is the amount of features you have, and you just calculate the distance, Euclidean distance between those coordinates. Okay, here an example. We can compare apples to orange using Tanimoto similarity. Actually, in Dutch, we don't say when you compare things that you shouldn't compare, we don't say comparing apples to orange, but we say comparing apples to pears. So therefore, I choose to compare apples, pears, and oranges. Here list of features for every one of these fruits. And you can see they have a few things in common. They're all edible, they're all fruits, but they're also different in a certain sense. So none of them is identical, their color is different, their grow climate is different, their shape is different, their skin is different, and so on. So here I made a matrix with the Tanimoto similarities. And so you can see apple to apple, well, that's 1. That's the maximum. Of course, it's fully similar to itself. Then here you can see apple to pear, that's 0.56. So what this corresponds to is 5 features out of 9 in total they have in common. So what this means is they're both fruit, they're both edible, they both grow in moderate climates, their main export country is China, their skin is smooth. And then there are 4 more features that they don't have in common, and this is the feature round, non-round, red, and green. If we compare apples and oranges, then we can see that actually just 3 out of in total 11 features are matching, and this means that our similarity metric is much lower. And actually, based on this, we can make we can see an interesting result, which I think intuitively is quite clear. Apples are a little bit closer to pears than they are to oranges. You could even say there are only 3 data points, so it's a little bit iffy, but one could even say apples and pears, they form a cluster. You can substitute them for each other in ways that you could not do for an orange. Let's say if you are making an apple cake, but you have only pears, this is fine. If you have oranges, that's not fine. And this is reflected in this quantitative similarity metrics. One more thing to talk about in this similarity part, and this is the concept of a chemical space or chemical spaces. What's a chemical space? It's an abstract concept that's basically that means all possible structures that exist under given criteria. So very typical will be something like synthesizable chemical space in under some drug-like parameters. So maximum amount of heavy atoms, maybe some Lipinski-like cutoffs, limits on the synthesizability, such things. And a typical chemical space, as we imagine it, is very, very large. Actually, the estimations differ many, many orders of magnitude. It really depends on where you put your cutoffs and whether it's 20 heavy atoms or 50. Actually, it really explodes. So this is why this number is fairly different. I won't go into this too deeply, but typically, if you want to visualize something like this, we can use dimension reduction techniques. For example, common linear one, that's PCA, which actually it shouldn't be used for binary fingerprints, but it works quite well nonetheless. Or also, as you've probably seen in the literature, there is Disney, which is like a stochastic method, which is more used for visualization, but it can accentuate the clusters which are present in the data quite well. Finally, why do I talk about chemical space? It's because it forms a bridge between the 2 parts of this lecture here. So the exploration of chemical space, moving around in chemical space, this is something like a molecular optimization. So here is just a visualization of what do these representations look like. Of course, this is not a whole chemical space. It's just a few thousand molecules. It's a data set I have where 1/2 are vanilloid receptor modulators, and the other are glucocorticoids. You can see here PCA linear method versus Disney. Natural clustering here is much more accentuated. There's an advantage, but the disadvantage is, well, it's a visualization method. It's very dependent. This is very dependent on its input settings. So actually, we cannot use this if down the line we would want to do classification or something like this. Okay. Oh yeah, actually, one more thing to mention about exploring chemical space. So there are actually there are some concrete ways how to do this. And this is molecular interpolation. So this is one thing which certainly I should mention, because the leader of my research group is one of the developers of this software. So what is done here is a mutation-based path between two different molecules. And in this way, the idea is that you have a nice clean path through chemical space. There are also a bit more contemporary methods. For example, using variational autoencoders, it's possible to, in a more or less continuous way, move between molecules. So you can see here, for example, in the latent space, it's sampled molecules around ibuprofen at a given distance. And you can see here, as the distance differs, you get further and further and more crazy variations. But actually, as you get quite close, you get quite nearby molecules. And let's say if you would want to find some analogs of ibuprofen that don't have some property that we don't like about it, but still is like a nice NSAID, nice COX inhibitor, probably searching in this region will be more fruitful than just randomly searching in some chemical database of some vendor that we can find. Now let's move to part 2, molecular optimization, and this is making use of similarity to get our properties of interest optimized. Yeah, as I wrote here as well, what we want to do in this task is very simple. We want to enhance our desired properties and diminish our undesired properties, and we of similar molecules. And actually, a typical molecular optimization, we don't necessarily even need similarity metrics for it or models for it. SAR, as it has been done since 1940s, 1950s, this is already a process of optimization. In that case, it's just people at the blackboard who are thinking, what if we put a methyl here? What if we remove this methoxy group and then measure it and investigate what's happening? So this is as well something like a guided optimization, very rudimentary guided optimization. So what are some properties to optimize? I will give a whole list of things we can optimize, but because it's a drug design workshop here, of course, the ones we care about the most are pharmacokinetics and pharmacodynamics. So first, what's pharmacokinetics? The classic abbreviation ADME plus sometimes T added to it. It basically means how the drug is moving through the body, what our body is doing to it, and this means that we need a molecule that can get absorbed, and this means that it needs to be soluble, it needs to be bioavailable. We need to have a correct distribution. For example, if we make a drug that we think is antidepressant, it needs to distribute itself to brain tissue, it needs to cross the blood-brain barrier, and so on. And also we need to care about metabolism, because after all, a drug is a foreign agent to a body. The body will try to destroy it to make sure that it's not harmed. And we have to make sure that our active drug doesn't get destroyed. And we also have to make sure that the metabolites themselves are not dangerous, that they are not associated with side effects, accumulation, such things. And then finally, there is the elimination. In the end, the drug has to be in the body only for a certain amount of time. Ideally, we would want to have constant plasma concentration over time, but in practice, you will have something like peaked behavior. You will have half time of elimination, something like this. And depending on what kind of drug we have, also this is a parameter which can be very different. For example, when it comes to chronic disease like chronic pain or schizophrenia or something like this, effectively we want to have a constant effect long over time. When it comes to something short term, something very acute like severe pain treatment or poison treatment or something like this, we rather want to have the opposite behavior. For example, if we want to do anesthesia, we want someone to be anesthetized only for a short time so we can do our operation and then they should get back to normal as soon as possible. And then finally, there is also this T factor toxicity, and this is usually explained by way of off-target behavior. So let's say if we have some nice, I don't know, kinase inhibitor or something like this, perhaps at the same time, it's also a HERG inhibitor and it will cause heart valve related problems. So we need to find some way to deal with this. And actually, because there are so many ways by which toxicity can occur, actually, this is a fairly difficult thing to model. Okay, the second bit that we care about, that's the pharmacodynamics. Dynamics as in thermodynamics, and this means that's the quantification of how well a drug is working at its target or at, let's say, the thing it's supposed to do. You can even calculate this very general. Also includes how many points are we reducing on the depression scale or something like this. What belongs to this domain, binding energy, activity at some assay, fluorescent assay, radioligand assay, something like this. Typical ways in which to express this are these four here. So that's the concept of inhibition, the binding consent, effective concentration at which we have 50% of our maximal outcome, inhibitor concentration 50% as well. We can even, outside of the biological medicinal chemistry domain, we can talk about host-guest affinity more generally. So for example, when it comes to small molecules inside of somewhat bigger guests that are still not proteins or nucleic acids or something like this, actually we can, that's another thing that we can optimize depending on what's our target, of course. For example, in the chemosensor case, that's something we would also care about. Then another important property to optimize, that's the synthesizability. The cost of production as well. Those two, of course, are related because the more difficult something is to synthesize, the more it will cost. Still, the golden standard, best method to do this, that's expert assessment. If you know some medicinal, rather organic chemist or you're organic chemist yourself, often to judge it yourself is simply the easiest. There are also some models that exist to estimate this. They are not perfect. The most commonly used one is SA score. There are a few more, RA score. In our group, we have one called SIBA. And they're quite good. Definitely, they are statistically, they are more or less correct about big data sets. It's actually quite a good way to filter them. And then finally, also, there is the cost of production. So ideally, if you are working with simple chemistry or in some very specific area of chemistry you know very well, then the cost of production in principle is no problem. Let's say it's the cost of your chemist's time plus the cost of your precursors. But if we do really deep de novo design, then often we don't know. They're not in any catalog somewhere. They have to be custom synthesized. And this means we need to do price prediction. And actually for this, we can build QSAR models as well. There is relatively good price data available. This has sometimes been called, instead of QSAR, QS $R, where the $ replaces activity. Then some more properties to optimize are general physical chemical properties. And actually, okay, why do we want to optimize them? Actually, a lot of these simple makes a good drug. What properties should a drug have? They are this type of physical chemical properties. So this is things like there is a maximal molecular weight, there is a lipophilicity, range of interest. For example, there can be maximum amount of hydrogen bond donors or acceptors in there. Then again, related concept, another thing we can optimize are steric properties, spatial properties. For example, if you are designing a drug, besides the selective interactions we want to make, well, we also care just about basic space complementarity to the binding site. If there is some cavity somewhere that we have not made use of, well, this is low-hanging fruit energetically to have something with nice space complementarity. Again, something that can be used outside of the medicinal chemistry domain. For example, if we are designing metal organic frameworks, if we have these entities like zeolites where you have these very big cavities, or if you want to design some molecule that can fit in Buckminster fullerene or something like this. Again, we really care about the steric and spatial properties. We want our molecules in that case to be spherical with the right radius. Now, I just gave a list of a huge amount of things that we can optimize a huge amount of parameters that are important in molecular optimization. And what that strongly suggests, what that strongly implies is molecular optimization is really a multi-parameter optimization. So there are several things which you are trying to optimize at the same time. So there are some classic examples here. Already I mentioned there is the Lipinski rule of 5. It's 4 rules of thumb. I think there is a certain log P criterion, there is a molecular weight criterion, there is a hydrogen bond donor criterion, hydrogen bond acceptor criterion, something like this. Then these type of rules, actually, they can be summarized into one value, which is very often used, especially by machine learning people. It's called QED, quantitative estimation of drug likeness, but it's actually just a simple formula which scores how well all these parameters are within a given range. Then when we are doing multi-parameter optimization, often the things we are optimizing, they are correlated and not necessarily positively. But one example of a positive correlation is as we get our molecule more lipophilic, in general, our activity will increase. But this activity increase is a false increase, because this means that, for example, if we are doing hit-to-lead optimization, if we are adding on, let's say, aromatic rings, we increase our lipophilicity, our binding values will be better, but actually our optimization space will become much smaller, because then we get outside of the range of what is typical for a drug that will be well absorbed in humans. So this means that despite that they are correlated, it's still something to consider, that we should try to control against. Yeah, and in general, actually, many of these descriptors, they are really correlated. So for example, if you have random chemical matter, well, the molecular weight is going to be correlated with the amount of rings. It's going to be correlated with the amount of heteroatoms, and therefore with the amount of hydrogen bond donors and acceptors. And this means that often there is often a lot of redundancy in there, because they are simply correlated. So it's one thing to keep in mind when selecting what are the features we care about. Well, are there not any underlying relations between them? Okay, as I remarked in the earlier slide, molecular optimization, it's really, you can conceive of it as a movement through chemical space. This is directed by feedback. In the classical medicinal chemistry way, this feedback is simply measurements. You predict a few compounds, you measure them, you follow the path that was useful. In the case of, in a more contemporary case, the first feedback we get is models. So we can do, I will talk about this in the next slide, but we have various ways to guess what we think the activity is going to be before we do the measurement. There is an assumption of smooth path through chemical space, but actually this activity landscape, as we call it, it's almost never continuous, typically either discontinuous or heterogeneous. So we will meet walls and valleys in this landscape, which I will very shortly. What are some ways, if you want to optimize directed by feedback using models, what are some ways to do it? So three major ways in which this can be done, from, let's say, a bit easier to a bit more difficult, at least from the point of view of computational time. We can build QSAR models and predict some simple properties we care about. The advantage of this is that it scales very nicely. We can do it on millions, even billions of compounds if you want. But there are certain limitations. You will see in the lecture about QSAR today, where they lie and how we can quantify them. And then there is the structure-based suite of ways to estimate this. A typical way to do it is docking. We can use this for data structure-based theory. So this means, does it really fit in the receptor? Does it really make the interactions that we were hoping for? Didn't the binding mode flip or something like this? We can use it as well for ranking. So when we do docking, typically we will get a docking score that will say how likely a molecule is to bind. Actually, in practice, individual docking scores, they are very unreliable, but they're still good enough to have a bit of enrichment. So for example, if I dock 100 million compounds, the best 100, they are going to be quite good. If I want to rank 2 compounds versus each other, the docking score is not going to be so meaningful. If you want to get a little bit more meaningful information, we can use a bit more involved techniques. For example, nowadays, what's commonly done is FEP, free energy perturbation, and there the scores that come out of this, they are a little bit more reliable, but this comes at the cost of much higher computational time. And also with these expensive techniques, still, we need to have a well-behaving system. We need to work in a certain chemical space. So even with those, if you use them for ranking and scoring, this is something that comes with given risks. Okay, I think I have 2 more things. Yeah, I have 2 more things to mention. And these are these optimization discontinuities. So when we are moving through chemical space, we can hit a wall. And what this means is that we have a sudden huge increase or decrease of activity. And this is called activity cliff. So we can see here these molecules, which from the point of view of similarity, let's say if you calculate the Tanimoto similarity between those 2 using ECFP fingerprint, let's say, well, they will be scored as very close, depending on the settings of the fingerprint, maybe 0.8 or 0.9. But we can see there is 2 orders of magnitude of difference in this pKi. Here again, there is a very simple flip of this functionality, and we can see the difference of pKi is more than 3. So this means that it's actually 3 orders of magnitude different. And this is often a problem of more simple QSAR similarity-based approaches, that these type of effects, they cannot be so easily anticipated, or rather the features which we choose, they are not able to capture such effects. And these effects, actually, they can be really subtle. For example, what's very famous in medicinal chemistry is the magic methyl effect. And what this basically means is when you add just a methyl group, just a carbon, you have huge increase or decrease of activity. Here again, you can see there is almost 3 orders of magnitude difference. And usually the way in which this is intuitively explained is that maybe there's some very small subcavity somewhere, lipophilic subcavity somewhere, and the method is in there in just the right way. It forces the molecule to take the right conformation that corresponds to binding. So even such small things, they can actually make a very big difference. Then the second thing which I wanted to mention is bioisostatic replacement. So this is one strategy with which we can move a little bit in a little bit parallel way outside of, let's say, normal similarity path. So what's a bioisostere? This is a chemical substructure that in some cases can be substituted for another while retaining the same biological activity. So here you can see a typical example, amide group, very common. It can be substituted by these other functional groups while in some cases retaining activity. And actually sometimes with this bioisosteric replacement, what you can do is you go from a amide, which normally likes to take a transoid conformation. So this means that this here is in one line, let's say. You can force it into a cisoid conformation. So it would be like this. Basically, the outgoing bond would be where is the hydrogen here. If we take a triazole, this one here, actually we force it to take this cisoid conformation. And if we see, for example, in the crystal structure that we have a cisoid conformation, we can we don't have to make the cost to having unfavorable conformation by locking it in and using the bioisostere that takes this specific form. Then one more thing to mention is the concept of the concept of scaffolds, and I mention it because this is another way to break through the similarity wall. This is usually described as a molecular core, something you decorate with substituents. So here in this case, the scaffold, this is Viagra, this is sildenafil. The scaffold would be this ring system with the substituents here being methyl, propyl, ethoxy, and methyl. A scaffold hopping, which, well, depending on who you ask, it has a little bit different meaning, but the core meaning is you replace the core structure, but you retain the activity. And in this way, you can find actives which are dissimilar from the fingerprint similarity point of view, but similar from the pharmacological point of view. And here on the right, you can see vardenafil, and this is a very good example of very fruitful scaffold hopping. So Viagra got marketed and became a billion-dollar drug. And one other firm, I forgot if it's, I forgot which one it was, maybe GSK, but don't quote me on it. Perhaps they had a very similar research program, or perhaps they decided works very well, they're earning a lot of money, so we make our Me Too drug, and we replace it with a heterocycle that is very similar electronically. There are more or less the same substituents, methyl, propyl, ethoxy, and okay, here ethyl instead of methyl, and here it's a successful scaffold hop. The drug works as well. It got marketed, and okay, Viagra made in its top years 2 or 3 billion per year. I think vardenafil was making some hundreds of millions, but that's still quite a good payoff. Then more generally about scaffolds, linkers, and substituents, there is a concept of the Markov structure, and this is a generic way to define molecules. And let's say often in these structures you can enumerate a huge amount of molecules. This is from the Xanax patent, and actually this whole defined space, let's say this scaffold with its various substituents on there, it defines in total 100 million molecules. Why do I mention this? Because that's one more thing that we like to optimize: patentability and novelty. If we have a drug design program, in the end, we want to lead to successful patented drug, and this means that we need to find ways to get outside of patent space. And bioisostatic replacement, scaffold hopping, that's one way to do it. So this concludes, I went 2 minutes over time, but this concludes the lecture. So let's think about the take-home message. If you want to optimize a molecule in the direction we want, if you want to get nice actives, no toxicity, good properties, we need very good quantitative similarity metrics. To have them good, we need to pick them in really meaningful way, which is really complicated tasks. And then finally, these features and this similarity can form the basis of more advanced modeling, which we will be talking about more today. Thank you. Yeah, of course.
You mentioned many approaches to make good drug. What is, from your experience, better combination? From what to start?
So I was trained as a synthetic and medicinal chemist. And this means that from my point of view, I have very coarse approach to this. So good activity comes first for me. So let's say if you get a nice ligand with very clear characterized activity, the orthogonal assays are fine, maybe you have a crystal structure. The other stuff you can typically optimize later. So if there is a log P problem, if there is a toxicity problem, something like this, it is a bit easier to fix than the alternative, where the alternative would be you have perfect properties, it dissolves perfectly, everything is fine, but the potency is really low. And actually, sometimes it happens that I'm working with collaborators, they want to find new ligand at this or that target, and they want to optimize everything at once. So they will say, okay, here is your list of molecules, but this one, we put it in our model and we don't like the log S so much or something. So then, okay, often we can make the decision, let's first see if it's to begin with, and then let's optimize the others. But on the same at the same time, we cannot send nonsense to chemists. So if it's unsynthesizable, if the properties are truly too crazy, we cannot do that. So we still have to keep these things in mind.
Would you say that the use of bioisostatic is more likely to be useful in a biological laboratory than classical chemicals? Or
the short answer would be yes, because after all, they're called bioisostates and not ordinary isostates. And actually, when you are doing a non-biological project, so for example, you are doing something with materials, let's say you're making dye compounds or something like this, actually you can use just ordinary isostates. And what this means is that they are electronically analogous. And this means that, let's say, we don't have to be limited by what a protein recognizes how, or what a nucleic acid recognizes how, or whatever. But let's say we can use a more coarse, simple approach. So I would say outside of the biological domain, it's actually a little bit easier.
No further questions? Let's thank Wim and invite Olena.
Thank you.
Can people on Zoom hear me?
Not yet.
Not yet. Is this better? Это не важно. Okay, so yeah, it should be working now.
It would be better if you write another line because it's still sound from the presentation.
Okay. Okay, so we'll wait for another minute because we have some sound delay.
Yeah.
I think the minute is gone. It's like,
Yeah, I could. Yeah, it's okay.
great. Okay, so now we can start and we'll go to something a bit different. And today I want to talk with you about natural products and their application in drug design. But before I start, I'm going to introduce myself. I'm a postdoc at IOCB Prague, at Tomáš Pluskal group. And what our group does is basically combines experimental and computational approaches for bioactive molecule discovery. So we have lots of people doing very interesting experimental stuff, and we maintain 2 bioinformatics software. One of them is MZMine and other is TransExpress. So after this brief introduction, let's talk what are natural products. So as you know, during the course of evolution, multiple species evolved, and all living organisms, they have some chemical processes happening inside of them that they need to survive. So any compound that is produced during these chemical processes inside living organisms is natural product. And when we speak about natural products, we can roughly split them into 2 main categories. Those of primary metabolites, those are usually simpler, they are similar across many organisms, and they frequently do not carry pharmacological activity. And then we have secondary metabolites, which are sort of non-essential specialized things like pheromones and toxins that organisms use for protective purposes, like to scare away predators, stuff like that. And because they were developed for these specific purposes, they usually have some biological activity. And I mean, it was noticed quite a long time before. People were using plants and mushrooms with unusual properties to treat illnesses, ailments. You can see, so as early as 3000 BC already there were some Sumerian clay tablets with some herbal recipes. Then there is very well known Ebers papyrus, and in Middle Ages people continued to do that. And first like pharmacopoeias were used throughout the history, and many people still use herbal remedies. Like, I don't know who doesn't drink chamomile tea when they're stressed out, for example. So, but these uses were relatively simple. They were using plants as a whole. But since then, natural products became a very prominent source of drugs. But the roots were in these like ancient practices. And before we speak about very well-known drugs that were derived from natural products, maybe you recognize this structure and can tell me what is probably the most well-known natural product derived drug is. Yeah, of course. I never doubted you. So, and aspirin was obtained from the bark of the willow and later modified specifically to minimize side effects such as ulcers and stuff like that. But of course, aspirin is not the only one, not the only natural products derived drug. Other well-known examples include anti-cancer drug Taxol, which was obtained from the yew, and this also very well-known drug artemisinin, which is anti-malaria drug, very widely used, obtained from sweet wormwood. And of course, not only plants are used to obtain drugs. There are, of course, fungi, for example, again, old school, very well-known example of penicillin, but also immunosuppressant sporin and another immunosuppressant rapamycin. This was obtained from fungus, and this was obtained from Streptomyces and has actually a very interesting history of discovery, because the bacteria from which it was obtained was found on Rapa Nui Island under these statues, and it's quite amazing. Actually, you can go there and find the plague where, with the location where these species were isolated. So if we look at the statistics from 80s to 2090, we see that more than 30% originate from natural products. So they are either natural products derived, semi-synthetic derivatives, etc., etc. And it is very well explained because we have this huge variety across the kingdoms of structures, scaffolds. As you can see, the most well-studied is kingdom of plants. This is the data from the Dictionary of Natural Products. Bacterial, fungal kingdom and animal kingdom are a bit less studied, but still there are some very interesting compounds that were obtained from them. So classifying all this variety of structures is quite non-trivial task that already requires application of computational methods. I won't stop really on that. I just wanted to show you how this variety of structures sort of proliferates into a variety of classes, such as flavonoids, proponoids, etc., etc. And there are several methods like how to classify the natural products when you obtain them. But let's speak, so, okay, plants and fungi, and we have some compounds that are produced by them. But what makes these compounds unique? And in other words, how do they differ from the synthetic drugs that we were discussing before? And when we discuss that, we must again think about evolution and remember common knowledge that plants don't run, or in more fancy words, that natural products are structurally optimized by evolution to serve particular biological function, which basically results in more complicated, more, let's say, more 3D structures. If you look at the two examples of best-selling synthetic drugs, apixaban and ibrutinib here, you see that compared, for example, to natural product-derived drug voclosporin, they have much more flatter structure. They have lower molecular mass. Natural product-derived drugs mostly have higher molecular mass, not always, if we think, for example, about aspirin, but mostly they have higher molecular mass, more sp3 carbons, definitely less nitrogens and halogens, more acceptors and donors, lower lipophilicity, which is not always a good thing, but at the same time, they have this sort of more specific 3D rigid structures, which basically makes them privileged to interact with the 3D protein target. So this all looks amazing. We have variety of species, we have a variety of products produced from these species, and we have these products have huge potential to become drugs. However, in 2009, there was a huge trend of decline of natural product research in pharmaceutical companies. There were lots of publications. If it's an end of the era, if no more natural products driven research will happen. So here you can hopefully see it. So it's just the extract from the article. Lots of pharmaceutical companies like Eli Lilly and Bayer's, they simply closed their natural products R&D programs. However, not all pharmaceutical companies done that. And I would say that it's sort of unclear trend, because at the same time, concurrently with decline